Hello guys, welcome back to Dr. Hill's Academy. Today we'll be discussing an important aspect of metabolism known as Krebs cycle. Stay tuned. So all along, we've talked about the glycolysis, we have talked about the entry of glucose into the cytoplasm through the GLUT transporters, right? And then we talked about what? Glycolysis, where glucose, this is your glucose, was converted to what? Pyruvate. Then the pyruvate enters what? The mitochondria. You know, the conversion of glucose to pyruvate, that's glycolysis, occurs in the cytosol, right? Then the pyruvate now enters the mitochondria through what? Pyruvate transporter. Okay, so over here we have our big boy pyruvate. So let's illustrate that with our P for pyruvate. And then the pyruvate is converted to what? Acetyl CoA through a reaction known as what? Pyruvate oxidation. Okay, so the enzyme is pyruvate dehydrogenase. So there will be an oxidative decarboxylation of what? Pyruvate to form what? Acetyl CoA. Okay, this pyruvate dehydrogenase is a complex, so it's a pyruvate dehydrogenase complex consisting of three enzymes. So it's a multi-enzymatic reaction. All right. Now the acetyl CoA, which we formed over here, this is the acetyl CoA, would now enter what? The TCA cycle. Okay, and then it will combine with this molecule over here. So these are oxaloacetates, right? The oxaloacetates. Now, this is the first step of the reaction. It is what? Irreversible. All right. So your oxaloacetate will combine with what? Acetyl CoA to form what? Citrate. Okay, citrate. And the enzyme involved is what? Citrate synthase. So let me use the red. Citrate synthase. So this is your citrate synthase over here. So you can see the illustration. The citrate synthase is a sucker for love, okay? So it combines the acetyl CoA and oxaloacetate in a matrimony to form what? Citrate, right? Now let me tell you all the intermediates in citric acid cycle, or remember Krebs cycle is also known as what? Citric acid cycle, or what? Tricarboxylic acid cycle, right? So we have Krebs cycle, also known as citric acid cycle, also known as what? TCA cycle. And what does this TCA cycle aim at doing? It takes the acetyl CoA and then combines with what? Oxaloacetate to form a 6-carbon molecule known as what? Citrate. And then this citrate continues in the cycle, liberating off what? CO2 and what? Giving off what? Energy. So you remember all along we've aimed at what? Breaking down glucose to form what? Energy and CO2, right? That's correct. Now I want us to use the memory aid. Oh, can I keep some succinate for myself? So, oh, can I keep some succinate for myself? So these are the intermediates in what? The Krebs cycle. Now I'll replace them with their real names. For the O, we have what? Oxaloacetate, right? For the can, we have what? Citrate. Then the I, we have what? Isocitrate. Then for the keep, we have what? Alpha ketoglutarate. Okay, so we are interested in the K, right? Then for the sum, we have succinyl CoA. For the succinate, succinate is succinate, right? Then 4, we have what? Fumarate. Then myself, we have what? Malate. So you can see the intermediates are oxaloacetate, citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, and malate. Now let's go to the main gist of the reaction. The acetyl-CoA combines with what? Oxaloacetate in a holy matrimony to form what? Citrate. Now, the oxaloacetate is how many carbon atoms? 1, 2, 3, 4. Acetyl-CoA is how many? 1, 2, right? And then citrate is what? 1, 2, 3, 4, 5, 6. So basically, we brought the oxaloacetate and acetyl-CoA and condensed it, okay? So whenever we condense it, what should we give off? Water. That's correct. So we'll give off water. And then there's something you have to remember, that this acetyl-CoA has a what? CoA, which I represented with this over here. It has the word CoA. So the CoA goes off as well. Alright? So these are CoA. Right? That's coenzyme A. Okay? Now this citrate synthase is an enzyme that can be activated or inhibited by certain substances. Some of the activators of the citrate synthase are what? ADP. Right? So whenever we have low energy states, we want to what? Enter the Krebs cycle to produce what? More citrate and then the rest of the reaction in order to liberate what? Energy, right? Now, what would inhibit it? I'm sure you guessed it right. ATP would inhibit it. Yeah, because that's high energy state, right? NADH would also what? Inhibit it. Because you know NADH is produced during the what? Breakdown of what? Glucose and even throughout the what? Citric acid cycle. Alright? Now, another thing that can inhibit it is what? Its product, what? Citrate. So we have citrate over here. Citrate would what? Inhibit it. And then we also have another molecule down the line, which is what? Succinyl-CoA. So this succinyl-CoA would also inhibit it. Now, these are the inhibitors of the citrate synthase. Let's go over to the next reaction. This was reaction 1. Now, let's go over to reaction 2. If you notice, reaction 2 is what? Reversible. Now, I told you the reversible and irreversible reactions of glycolysis. Let me tell you the reversible and irreversible reactions of what? Krebs cycle. So, reactions 1, 3, 4 of Krebs cycle are what? Irreversible. Remember I told you 1, 3, 10 for glycolysis are irreversible. In this case, 1, 3, 4 are irreversible. So reaction 1 was irreversible. Now reaction 2 would be what? Reversible. Okay? So citrates will be converted to what? An isomer known as what? Isocitrate. And the enzyme that does this is this rat over here. So this rat is called what? Aconitase. Aconitase. Aconitase would catalyze reaction of what? Citrate to what? Isocitrate, which is just an isomer of citrate. That's why we still have our carbons intact. 5, 6. Okay? Now, there's something important about this enzyme. There is a substance found in rat poison called what? Fluoroacetate. This substance, fluoroacetate, is found where? In rat poison. Now, we want to kill these rats. Okay, would give fluoroacetate, and this fluoroacetate would be acted upon to form what? Fluoroacetyl-CoA. Okay, the fluoroacetyl-CoA would undergo the same reaction as the normal acetyl-CoA and then form what? Fluorocitrate. And the fluorocitrate would now bind to this enzyme. This enzyme will think, oh, this is my normal citrate, right? Then it binds to what? Fluorocitrate, and the fluorocitrate holds it bound, inhibiting the enzyme. Okay. So that's one inhibitor of this reaction. That's what our fluorocitrate. Okay. What else inhibits this enzyme? This enzyme can also be inhibited by something you all know, zinc. So zinc can inhibit this enzyme. Okay. Now, let me tell you something you all should know. In prostate cells, they have so much zinc. Okay, their mitochondria have what? Zinc transporters. Okay, so in the mitochondria of prostate cells, we have what? Zinc transporters. Okay, which transports zinc into the mitochondria, and then this zinc inhibits what? Aconitase. When aconitase is inhibited, what happens? Accumulation of what? Citrates in the prostate cells. So this citrate is now secreted onto what? The semen, alright, onto the sperm cells, and then it now helps in what? Sperm cell motility. Okay. Now, in cancer cells, maybe an individual has prostate cancer, what will happen? There is what? Mutation of this zinc transporter. And then zinc does not rush in. So there's low levels of what? Zinc in prostate cancer, which culminates in what? Low levels of what? Citrates. That's in what? Pathological states. That's your prostate cancer. Now we finished with this boy over here. And remember, this was our reaction 2. Let's move over to what? Reaction 3. So is reaction 3 irreversible or reversible? It's irreversible. I told you 1, 3, 4 are what's irreversible. So this is our reaction 3. What happens in reaction 3? Isocitrate is converted to what? Alpha ketoglutarate. Oh, let's check the number of carbon atoms. Remember, isocitrate was 6, right? So alpha ketoglutarate is 1, 2, 3, 4, 5. Whoa, we've lost the carbon. How do we lose carbon? As what? CO2. All right. So a carbon atom is lost as what? CO2. Right. This reaction is catalyzed by which enzyme? Can you guess? Remember, this was isocitrate. This is alpha ketoglutarate. Isocitrate what? Dehydrogenase. Okay. So we have our what? Isocitrate dehydrogenase. What did I tell you about dehydrogenase? I told you whenever we have a dehydrogenase, we are thinking what? NAD, right? So NAD is used, NAD plus, right? And then we have what? NADH. This NADH would eventually enter the what? Electron transport chain to liberate what? ATP, okay? Now, this our baby owl here, can you tell me things that activate it or inhibit it? First, let's look at what? The activators. Now, we have activators. What do you think would activate it? Tell me ADP, right? ADP. Whenever we have high ADP, what's our cell trying to say? It's trying to say, oh no, I don't have enough energy, right? So ADP indicates what? Low energy levels. So to activate this isocitrate dehydrogenase so that this reaction can continue and what? Generate what? Energy, right? What else do you think would activate this? You might not know, but let me tell you. Calcium. So your baby here, calcium, is produced, for instance, take for instance in skeletal muscle cells. So skeletal muscle cells produce what? Have high intracellular what? Calcium levels whenever they want to contract, right? And contraction requires energy, right? And then also cells that secrete hormones or other substances, they have to transport these vesicles towards the membrane. How do you think these vesicles are transported? Filaments help in the transport of these vesicles. And then this requires what? Energy. Okay? So whenever we have high calcium, it's telling the cell, okay, look, produce more energy, right? That's correct. So we've seen some activators of what? Isocitrate dehydrogenase. Now let's look at some inhibitors. What do you think would inhibit it? Please, please, please tell me what? ATP. ATP would inhibit this enzyme, isocitrate dehydrogenase. What else would inhibit it? Whoa. NADH. NADH indicates what? High energy levels. Remember, we've been forming NADH all along from glycolysis, and then pyruvate dehydrogenase, and now we form NADH. So NADH indicates what? High energy levels, and then it would what? Inhibit isocitrate dehydrogenase. So NADH. Boom! We are done with reaction what? 3. Now let's go over to reaction 4. Now we have our big boy over here. Big boy known as what? Alpha ketoglutarate what? Dehydrogenase. Please, if you don't know this enzyme, you don't know TCA cycle. This is alpha ketoglutarate dehydrogenase, right? This enzyme is very important. Okay, now listen carefully. Alpha ketoglutarate was formed earlier from what isocitrate dehydrogenase. Now, this big boy over here would what liberate a carbon atom in form of what? CO2. Okay, so we have our CO2 rushing out of the cell. All right. Now, this alpha ketoglutarate, you had alpha ketoglutarate dehydrogenase, right? So what do you expect? NAD, right? NAD plus will be converted to what? NADH. Okay. I want to tell you something about this enzyme also. This enzyme is similar to what? Pyruvate dehydrogenase complex. So this is alpha ketoglutarate dehydrogenase what? Complex. Consists of how many enzymes? Three, E1, E2, and E3. I already did a video on pyruvate dehydrogenase complex or oxidation of pyruvate, so you can better understand this enzyme as well as PDH complex. Okay. Now this enzyme has a subunit that transfers what? A-CoA. Come on, I told you that it's similar to pyruvate dehydrogenase complex. So you should expect what? CoA, right? So CoA is sent to it. And then this is our CoA here. Just like acetyl CoA, right? Correct. So we have what? Succinyl CoA. Okay. Now let's talk about some activators and inhibitors of this enzyme. Okay. Our alligator over here can be activated by what? ADP. Obviously, low energy state, right? What else? Calcium. Are you not happy that it's the same thing? So you don't need to bother your head. So just know that some activators activate this one over here, right? Now, how about the inhibitors? The inhibitor of this boy over here would be what? ATP, right? ATP would inhibit it. What else can inhibit it? NADH. What else can inhibit it? One more, apart from this one. One more. Succinyl CoA. Okay, so it's inhibited by what is products. Okay, so some sort of negative feedback, right? So succinyl CoA. All right. So these are what inhibitors. Something can go wrong in this pathway. What can go wrong? Some babies are born with deficiency of what? Alpha ketoglutarate dehydrogenase. Now, whenever there is deficiency, can you tell me what you would expect? High levels of what? Alpha ketoglutarate. So there will be what? High levels of what? Alpha ketoglutarate. What will happen? It will now spill into where? The urine. So in the urine, we will have what? Alpha ketoglutarate. Okay. What else would you expect? Now remember that this pathway continues what? The breakdown of what? Glucose. Remember we've finished with glycolysis, we finished with oxidation of pyruvate. Now we are talking about TCA cycle. And then this, when there's a break here, so there's deficiency of this enzyme, this pathway, this particular reaction is blocked. What would you expect? There'll be what? Accumulation of what? Previous intermediates. And then one of the remarkable things that accumulated is what? Pyruvate. But the pyruvate is now shunted into something else called what? Lactate. Remember we talked about it in a video on glycolysis, where I told you that lactate dehydrogenase has to come in to convert the pyruvate to lactate in order to free up what? NADH, right? Now, there'll be what? Increase in what? Lactate. Okay, so the lactate will be way high. That's the blood lactate. So the person will have what? Congenital what? Hyperlactatemia. The person will have what? Congenital hyperlactatemia. Okay. What else would you see? You see that the baby over here would come down with what? Hypotonia. Okay. So these are classical signs of what? Alpha ketoglutarate dehydrogenase what? Deficiency. So remember, since lactate is what? An acid, you would have what? Acidosis. Okay. Now we are good with reaction 4. We are progressing. Let's go over to reaction 5. So let's go over to what? Reaction 5. Now, this is reaction 5. Irreversible or reversible? Reversible, remember 1, 3, and what? 4 are irreversible. So this is what? Reversible. And then there's what? Conversion of succinate CoA to what? Succinate. Enzyme involved is what? Succinate thiokinase. Okay? It's a pretty small enzyme that doesn't have any problem. Succinate thiokinase. The only problem is this name. There are two names. You can also call it what? Succinate synthetase. All right. So either succinate thiokinase or what? Succinate synthetase. And it converts succinate CoA to what? Succinate. Boom. What are you thinking? CoA is leaving. So we have to remove this boy over here. They what? CoA. So let me use the black. So we have CoA being what? Given off. And then you had succinate synthetase. Are you not thinking of an ATP, right? But in this case, it's what? GTP, okay? So we'll have a GDP coming over and then giving off what? GTP, all right? So GTP is also an energy form, right? But then the GTP is now converted to what? Back to GDP by what? ADP, which takes up the phosphate and then forms ATP. So eventually we have what? Our ATP. Okay, so we just had to do it indirectly because of this our baby duck over here. So that's all about the enzyme. Let's go over to what? Reaction 6. We have our reaction 6. Irreversible, right? No, reversible. Okay, so succinate is converted to what? Fumarate. Now, our boy over here is sitting down on where? Inner mitochondrial membrane. Why? Because this enzyme sits on what? Inner mitochondrial membrane. Okay? This enzyme, well, you can call it complex 2 if you want. Well, but that's when it's involved in what? Electron transport chain. Because this enzyme is a part of the electron transport chain. Okay? But it's also a part of the Krebs cycle. You know, it's multidimensional. So this enzyme is called what? Can you guess? Succinate dehydrogenase. Yeah, succinate dehydrogenase. Okay, let me remove this 2 here. It makes the enzyme look weird. Okay. Let's put the 2 over here. No, it's thinking over here. Succinate dehydrogenase. Now, this enzyme converts succinate to what? Fumarate. You heard succinate dehydrogenase. What are you thinking? Don't tell me NAD. It's what? FAD. Okay. Well, FAD is required. Why is FAD used instead of NAD? Succinate converting to what? Fumarate does not liberate as much energy as some other dehydrogenases. Succinate dehydrogenase is not very strong, okay? That's why it's very weak, sitting down on the table, right? So it liberates little amount of energy that FAD can use and be satisfied, okay? So FAD come over here and then giving off what? FADH2, all right? So FAD is converted to what? FADH2. FADH2 can now enter what? Electron transport chain, right? Now, something can go wrong with this enzyme. When this enzyme is deficient, what happens? You know, this enzyme is called what? SDH. That's succinate what? Dehydrogenase. Succinate dehydrogenase consists of what? 4 isoforms. So we have A, succinate dehydrogenase A, we have that of what? B, we have C, and we have what? D. We inherit all these from our father and mother, so we have 2 of each, right? Now, succinate dehydrogenase A is the good boy over here. And then these other ones are bad boys that can be involved in what's cancer, okay? Tumor development. This succinate dehydrogenase A protects us against what? Tumors. So whenever there is what? Deficiency of this succinate dehydrogenase A, we can develop what? A tumor, all right? Because it protects us, and since there's no protection, this bad boy is over here now override it, okay? And then we have tumors, right? Now, for the B, C, and D, if there's a mutation in any of them, we can develop what? Tumors. One of the most common tumors that is developed from this is what? Pheochromocytoma. Pheochromocytoma, which is what? Tumor of what? Adrenal medulla. Okay, that's about this boy. Now let's go over to what? the second to the last reaction. This is what? Reaction 7. We're almost done, okay? So, reaction 7. Converting what? Fumarate to what? Malate. What's the name of this enzyme? Fumarase. Okay? Fumarase. Now, this fumarase catalyzes the conversion of what? Fumarate to malate. What is it doing over here? This is a fish, right? Being a fish, you expect what? Water to be involved. So it's adding water to the reaction. Okay. Now this step would have been a very easy walkover if not for the fact that there are deficiencies that could arise from this step. Now the fumarase can be what? Deficient. When this fumarase is deficient, what will happen? There will be accumulation of what? Fumarate. When fumarate is accumulated, it has been shown to be involved in some, pathological conditions like what? Uterine leiomyoma. Forget the medical jargon, it's called what? Fibroid, okay? Uterine fibroid. All right. Another one is what? Cutaneous leiomyoma. So basically, tumor of what? Areata pili. That's one of the most important ones clinically, right? Now, another thing that can occur is what? Renal cell carcinoma. Okay, so some tumors involving what? The kidneys. Now, that's all for what? Reaction 7. Let's go back to reaction 8. So, this reaction 8 is the last but not the least. Okay, we have malate dehydrogenase, which converts malate back to what? Oxaloacetate for the cycle to continue, right? Now, this malate dehydrogenase, being a dehydrogenase, I'm sure you would expect NAD plus to be converted to what? NADH. That's great. So let me write out the name of this enzyme for you. All right, that's malate dehydrogenase. All right. Now we are done with the 8 steps of what? TCA cycle. Let's just have a sneak peek into the energetics of the cycle. So before we can run the energetics, let me remind you something. One glucose molecule was forming what? 2 pyruvates. Remember our glycolysis, right? The 2 pyruvates now come to form what? 2 acetyl-CoA. So this reaction is for what? 1 acetyl-CoA. So whatever we are getting here will be multiplied by 2 for 1 glucose molecule. I'm sure you get it. So what I just said was that 1 glucose is eventually forming what? 2 acetyl-CoA. And this reaction was for 1 acetyl-CoA. So whatever we get, we can multiply by 2. So this reaction occurs what? Twice. Now let's count these molecules together. So we'll go to first one, reaction 3. What do we have? NADH. So from reaction 3, we have what? NADH. So to be what? 2 NADH. Right? Now, reaction 4, we have NADH, right? So we have reaction 4 giving off what? 2 NADH as well. Now, coming over to reaction 5, what do we have? ATP, right? We have 1 ATP, that's what? 2 ATP, eventually for 1 glucose. And then, over to reaction 6, we have 1 FADH2, that would be 2 FADH2, right? Then, over to reaction 8, we have what? 1 NADH, that's what? 2 NADH. Okay, so let's sum everything up. We would have 1, 2, 3, that's 3 times 2, that's 2, 4, 2, 4, 6, that's what? 6 NADH plus what? 2 FADH2, that's 2 FADH2 plus what? 2 ATP. 2 ATP. All right. So this is what we have for the Krebs cycle. So in the electron transport chain, 1 NADH gives what? 2.5 ATP. Okay. We have 1 NADH giving what? 2.5 ATP. And then one FADH2 gives off what? 1.5 ATP. So if you look at it, we have 6 NADH. So 6 times 2.5 would give us what? 15 ATP. And then we had 2 FADH2, that's 2 times 1.5 will give us what? 3 ATP. And then we had 2 ATP, right? So that's 2 ATP. Okay? So you would see that eventually you would have what? 15 plus 3, 18 plus 2, 20 ATP generated by Krebs cycle from one glucose molecule. So guys, we've come to the end of this lecture. I hope you enjoyed it. Do well to subscribe, like, put your comments below, and then share to your friends so they can benefit from this lecture. Thank you, and see you in the next lecture.